Welcome back to this next video, and in this video we are going to talk about the fork line method. This particular method, that is an alternative method to the Punnett square, and in this particular method, instead of showing the progenies in a square by combining the gametes systematically as we do in the Punnett square, in this particular method we carry them in a diagram of branching lines. This fork line method, it can predict the outcome of a cross involving two or more genes. This means that you cannot use this particular method for the monohybrid crosses. So when you talk about the fork line method, you will be focusing on crosses involving two or more genes. By that I mean you can use this for the dihybrid crosses, for the trihybrid crosses, or the multihybrid crosses. We will discuss the fork line method by taking an example of the dihybrid cross. Now, what dihybrid cross mean? The di mean two, that means you are focusing on two traits. The hybrid mean that each of these traits that have got two different alleles, and the cross mean you are actually crossing two parents, usually a male and a female, and they are going to combine their genetic information. In this particular example, we will be focusing on two of the traits of the seed that was used by Mendel, and we will be focusing on the shape of the seed, and we will be focusing on the color of the seed. Say, for example, if you cross a heterozygous round yellow plant to a heterozygous round yellow plant, now both of the parents they are of the same nature. They are heterozygous round yellow. That means that the trait round, the shape of the seed that you're talking about, that is in the heterozygous condition. The color of the seed that you're talking about, that is also in the heterozygous condition. What I mean by that? Heterozygous round mean that the genotype for this particular trait, that will be in the it will be having one dominant allele and it will be having one recessive allele. Now this particular capital R and the small r, that is actually showing you the first trait, which is the shape of the seed. The other trait, the color of the seed, that is actually represented by this capital Y and the small y. So if you talk about the genotype for the first trait, which is the shape of the seed, that is in the heterozygous form. That means that in this particular pair, one of the allele that is dominant, the other allele that is recessive. So in the pair, both of the alleles, they are different from each other. Therefore, we are using the term hetero. Hetero mean different. Similarly, when you talk about the color of the seed, one of the allele in this particular pair is the dominant allele, and the other member of this pair, that is the recessive allele. Therefore, you have one capital Y, you have one small y. That means that this particular pair is also in the heterozygous form. So this is what you mean by a heterozygous round yellow plant. The other parent, of course, is of the same nature. Heterozygous for the shape of the seed, and heterozygous for the color of the seed in terms of its genotype. When you talk about the fork line method, what you have to do is that, say for example, if you are talking about a dihybrid cross, first you have to partition them into two monohybrid crosses. If you are focusing on a trihybrid cross, that means that the three of the traits, you have to first make the monohybrid crosses for all of them, and after that you have to make the fork line method for it. Now let us discuss this particular example. If you talk about the shape of the seed, it comes in two varieties. One, the seed can come into the round shape. The seed, they are also available in the wrinkled shape. The round shape of the seed, that is a dominant trait. This means that it can express itself in the homozygous as well as in the heterozygous condition. If you talk about the wrinkled one, this is a recessive condition. That means it can only express itself in the homozygous form, when both of the alleles, they are small r and small r. The round, as that is a dominant one, it can express itself like the capital R, and also is the capital R and the small r. You can see an example over here that it is capital R and small r, but still the phenotype of this particular plant, the shape of the seed, that is round in nature. So the shape of the seed that come in two forms, the round and the wrinkled. Is the round that is dominant, we represent them with a capital R. Is the wrinkled that is a recessive, we express that with a small r. When you talk about the color of the seed, again it come in two varieties. The seed can be yellow in color, the seed can be green in color. Is the yellow color that is dominant, the green is the recessive one, therefore we represent the yellow allele with a capital Y and the green allele with a small y. So first we have to make the monohybrid crosses. If you talk about this parent, so this means we are focusing on the first pair of the alleles, the capital R and the small r. Same will be for this particular parent, the capital R and the small r. So when you make the gametes for the Punnett, for the monohybrid crosses, what you have to do is that you have to separate these members from each other. So one of the gamete can contain the capital R, the other gamete can contain the small r. So these are the gametes of one parent. If you talk about the other parent, again it is having a capital R and the small r. That means that the other parent, one of the gamete can contain the capital R, the other gamete can contain the small r. Now you have to cross them. This capital R, this gamete from this parent can combine with this gamete from this parent, giving you capital R and capital R. This gamete, it can combine with this particular gamete, so you are actually having the capital R and the small r. This capital R can combine with the small r, giving you capital R and small r. And this R, it can combine with this particular gamete, giving you the small r and the small r. Now when you talk about the genotypes in this particular case, if you see the capital R and capital R, they are actually making 1/4 of all of the offsprings. Because this is only one, so out of 4 it is represented by 1/4. If you talk about the capital R and the small r, this is 1/4, this is 1/4. That means that the capital R and the small r collectively they are making half of the offsprings. They are making 50% of the offspring. If you talk about this one, this is actually making again 1/4 of all of the offsprings. So what you can get from this particular monohybrid cross is that 1/4 of them, they are going to be capital R and capital R. Half of them, because out of 4, if 2 are capital R and small r, that means that half of them that will be having a genotype of capital R and the small r. And if you talk about this one, again, they are making 1/4 of all of the offsprings. If you talk about the color of the seed, again, if you can see this one, this particular parent, it can have gametes having the capital Y, having the gametes with the small y. Again, this second parent, if you talk about this one, the gametes can have the capital Y, the gametes, they can have the small y. Now you have to cross them. One gamete from this parent is going to combine with the other gamete from this parent, giving you capital Y and capital Y. This gamete can combine from this parent to the gamete of this parent, giving you the capital Y and the small y. Similarly, this capital Y, it can combine with the small y, and this small y from this parent, it can combine with the small y from this particular parent. Now, if you talk about this one, the capital Y and the capital Y, they are actually making 1/4 of all of the offsprings, because only one is there. If you talk about the capital Y and the small y, they are making half of the offsprings. They are having a genotype like this, and again 1/4 of the offsprings, they are actually having the genotype like small y and small y. So now you have made the monohybrid crosses, and you know the probabilities of the genotypes of the offspring. Now let us make the fork line method for this now. Now the first thing you have to do is that if you talk about this one, if you talk about the capital R and the capital R, they are actually making 1/4 of the total offspring. So 1/4, they will be the capital R and the capital R. Now what you have to do is that you have to make a fork like this. You have to make 3 lines first, this one, and you have to cross this particular genotype with all of the genotypes that you are expecting for the color. So if you see this one, 1/4 of them they are the capital Y and the capital Y. Half of them they are the capital Y and the small y, and 1/4 of them they are the small y and the small y. If you talk about this monohybrid cross, we are getting the 1/4 is the capital Y, half of them is the capital Y and the small y, and 1/4 of them is the small y and the small y. Now you have to cross them now. So if you cross this 1 into 1, that will be 1. If you cross this 4 into 4, that is going to be 16. And their genotypes will be, you have the capital R and the capital R from this particular parent, the capital Y and the capital Y from this particular parent. So this means that out of the total 16 that you make in the Punnett square, out of the 16, one of them that will be the capital R, capital Y and the capital Y. If you cross the second one, like say for example, if you cross this with this one, so 1 into 1 that will be 1, 4 into 2 that will be 8, and their genotype will be the capital R and the capital Y and the small y. That means that if out of 8 they are 1, that means out of 16 they will be 2. So 2 out of the 16, their genotypes will be the capital R and the capital Y and the small y. If you cross this with this one, what you are going to get is 1 into 1 that will be 1, 4 into 4 that will be 16, the capital R from here, the small y and the small y from here. That means that out of 16, one of the offspring that will be having a genotype like this one. So this is for the first one. Now you have to repeat this for all of the values that you have got from the monohybrid crosses. So we have done this one. Now let us focus on this one, the 1/2 of R and R. So what you have that you have half of the offsprings with the genotype like the capital R and the small r, and you have to cross them with all of the values that you have got for the monohybrid crosses from the other trait. So it will be 1/4 again, that will be the capital Y, half of them that will be the capital Y and the small y, 1/4 of them that will be the small y and the small y. Now again you have to cross them. So 1 into 1 that will be 1, 2 into 4 that will be 8. That means 1 out of 8 that will be having a genotype like the capital R, small r, capital Y and capital Y. So if out of 8 they are 1, so out of 16 that will be 2 in number, and their genotype that will be the capital R, small r, capital Y and the capital Y. Now you have to do it for the other one as well. So 1 into 1 that will be 1, 2 into 2 that will be 4. That means the genotype will be capital R, small r, the capital Y and the small y. So if out of 4 they are 1, that means that out of 16 they will be 4. And their genotype will be the capital R, small r, the capital Y and the small y. If you cross this one, this one, so 1 into 1 that will be 1, 2 into 4 that will be 8. And their genotype will be the capital R, small y and the small y. And out of 8 they are 1, out of 16 that will be 2 in number. So 2 out of 16 the offspring they will be having a genotype like this one. Now if you talk about the third one, like if you talk about this one, 1/4 of them they will be actually having 1/4 of them they are the small r and the small r, and you have to cross them with the 3 of the values that you have got from the monohybrid cross of the color of the seed. So it will be 1/4 will be the capital Y, half of them they are the capital Y and the small y, and 1/4 of them they are the small y and the small y. Now if you cross them, this means that first you have to cross this with this one. So 1 into 1 that will be 1, 4 into 4 that will be 16, and their genotypes, that will be the small r, capital Y, and the capital Y. So out of 16, the genotypes of these kind of the offsprings, they will be like small r, capital Y, and the capital Y. If you cross this with this one, what you are going to have is 1 into 1, 1, 4 into 2, that will be 8. The genotype will be small r, the capital Y, and the small y. So if out of 8 they are 1, so out of 16 that will be 2. So 2 out of 16, the genotypes will be small r, capital Y, and the small y. Cross this with this one, we are going to get is 1, 4 into 4, that will be 16. Their genotypes will be small r, small y, and small y. That means out of 16, one of the offspring that will be having a genotype of small r, small y, and the small y. Now, if you talk about this one, if you look at the phenotypes of the offspring, this particular one, if this is capital R, small y, and small y, that means this will be round yellow in nature. If you talk about the phenotype, because both of the alleles for both of the traits, they are in their dominant form. If you talk about this one, again, this will be round, because both of the alleles, they are present for the round trait. The capital Y is that is dominant over the small y. So of course, this kind of the offspring, that will be round yellow in nature. This is not W, that will be round yellow, and this will also be the round yellow in nature. If you talk about this one, again, this will be capital R, that means the shape of the seed, that will be round, but at both of the alleles, they are for the green color, the small y and the small y, so their color, that will be green. If you talk about this one, again, this is round because the capital R, that is dominant over the small r, capital Y, so this will be a round yellow in nature. If you talk about this one, this is the capital R is dominant over the small r, the capital Y is dominant over the small y. So the this particular offspring, that will be round, and that will be yellow. If you talk about this one, again, the capital R is dominant over the small r, so it will be round, but as both of the allele, they are present for the recessive color, so the color of these particular offspring, that will be green. If you talk about this one, this R and R, that is actually showing you a wrinkled shape. The capital Y and the capital Y, that is showing you the yellow color. This, again, the small r and the small r, that is showing you the wrinkled shape, but as the capital Y, that is dominant over the small y, so the color, that will be yellow. If you talk about this one, you have got both of the traits in their homozygous form, and both of them, they are for the recessive. So the this particular offspring, that will be wrinkled, and this will be green. So if you are going to classify them, so the first kind of the offspring that we are getting, they are the round green, not the round yellow. Let us count the round yellow first. How many round yellow they are? They are out of 16. So if you talk about this one, this is round yellow. This is one round yellow. This is round yellow, but there are two of them. So we have got a total of 3. If you talk about this one, they are 2 in number. That means we have 5. This is a round yellow, and there are 4 of them. That means we have a total of 9 round yellow over here. So the number of the round yellow offspring, that will be 9. If you talk about the round and the green one, how many of them they will be there? So let us count the round green. This is round green, 1 of them. This is round green, there are 2 of them. So 1 and 2, they are making 3. So you have got 3 round and green. Let us talk about the wrinkled yellow. How many of the wrinkled yellow they are there? So if you count the wrinkled yellow, so this is wrinkled yellow, 1 wrinkled yellow. You have got another 2 wrinkled yellow, so a total of 3 of the offspring out of the 16, they will be wrinkled yellow. And if you talk about the wrinkled green, there is only 1 of the offspring, with this one, the small r, and the small y, and the small y, which are actually having the wrinkled green phenotype. To understand the Punnett square, you should see my video, and I will share the links in the description.